Hello, everyone, and thank you for tuning in into this new episode of Beats Research Radio, a podcast and YouTube channel that aims to disseminate science and research to the community. My name is Deyanira, a PhD student in biochemistry at the University of Ottawa, and I will be your host in today's episode. Today, we have the great pleasure to be joined by Dr. Brian Aguado, an assistant professor at the University of California, San Diego, where he leads the Aguado Biomaterials Research Group, focused on developing precision biomaterials to investigate sex differences in cardiovascular disease. Dr. Aguado completed his bachelor's degree in biomedical engineering at Stanford University, followed by an MS and PhD in biomedical engineering from Northwestern University. He then completed his postdoctoral training in the Department of Chemical and Biological Engineering at the University of Colorado Boulder. His outstanding work has earned him numerous honors, including the prestigious National Institute of Health Director's New Innovator Award, the Chan Zuckerberg Initiative Science Diversity Leadership Award, and the National Science Foundation Career Award. Beyond his scientific contributions, Dr. Aguado is also a passionate advocate for equity and inclusion in STEM. He is a co-founder in Latinx in BME, a vibrant community that supports Latinx biomedical engineers and scientists through their careers. Thank you so much for joining us, Dr. Aguado.
Thank you so much for having me, Deyanira. It's so good to see you, and it's great to see you since my visit in Ottawa.
Yeah,
And I'm glad to join you here today.
thank you so much. So I would like to start this conversation with a question about your research and your lab. So your lab focuses on sex-specific responses to cardiovascular disease using biomaterials. And so could you explain our community why it is important to study sex as a biological variable in this context, and also how it has changed the way that we understand disease?
Absolutely. So, um, studying sex as a biological variable is really fundamental to improving how we understand, treat, diagnose, and ultimately cure cardiovascular diseases. And for so long, I feel like most biomedical research has been conducted using male biased approaches. For example, using an unknown population of cells. They could be mixed population of male and female cells, or just exclusively using male cells. It could be using male animals or male-centric clinical data, right? Because there's this long-standing mentality that female hormones could impact your experimental results, and that leaves a gap in understanding female-specific physiology, right? And again, thinking more clinically, men and women experience cardiovascular disease very differently. So, for example, my laboratory, we're very interested in aortic valve stenosis. Aortic valve disease, it typically in males develops more calcification in the aortic valve relative to females that experience more fibrosis in the aortic valve. And these aren't just minor differences, they really point to fundamentally distinct disease mechanisms. So, my lab, we're trying to figure out these sex dependent mechanisms to define how aortic valve stenosis progression depends as a function of sex. You mentioned that my laboratory, we focus on developing precision biomaterials. So we use different types of hydrogel biomaterials to recreate the valve tissue environment of of the aortic valve tissue. And we essentially use these material platforms to compare how male and female cells respond to the material stiffness of our culture platform. And we found that biological sex can influence everything essentially, from how cells sense the material stiffness to how they activate in response to biochemical factors like hormones or inflammatory proteins. In addition, we've also found that most strikingly, sex chromosome biology can drive sex specific cellular responses. So we know that two X chromosomes in female cells can actually drive these pro-fibrotic signaling networks inside of cells to drive fibrosis in females. But we also know now that the Y chromosome is also involved in driving osteoblast-like differentiation in the valve and driving this aortic calcification. So again, I think that it's so important to consider sex as a biological variable in study design, because if these kind of insights would be missed entirely in a sex agnostic study, right? So I think that studying sex differences really completely changes how we think about the pathogenesis of different diseases and how we can design more effective treatments for patients based on sex.
Thank you so much for explaining this in this way. I think it's really useful for us as researcher to also include this in our own research and to learn more about why it is important. And personally, I consider that is really useful and everyone is benefit from this kind of research because this can help to have more a specific treatment for everyone because not only one treatment will fit everyone, I think.
Exactly. That's exactly right.
Yeah, thank you. Thank you so much for these answers. I would like to continue with our second question, which is one of the major challenges in biomedical research is designing experiments that truly reflect sex differences. What have been some of the biggest obstacles you may have encountered during your research?
I think that so there's a number of different examples or obstacles rather. I think that one of the biggest challenges that I've encountered in my research and my laboratory has encountered is really having access to sex-identified primary cells and tissues. So when I first got started in this field, um, the standard protocol in my postdoc lab was to source pig valves without knowing the sex of the animal, right? We just got a box of hearts and we just used those cells and tissues in a sex agnostic way. But when I learned that valve disease had such striking sex differences, I took it upon myself to actually call the company and say, hey, can you ship me separate boxes of male and female hearts, right? And again, these are pig hearts when we first got started with this research. So when I started to sex separate the samples and I started to observe sex differences in cellular behaviors, I knew that this was kind of a turning point for me to realize that it wasn't so much a challenge per se; it was just more of like inertia, right, and like getting started. And I think that once you realize that there are certain protocols in your laboratory that are kind of established, you really have to take it upon yourself to change those habits and kind of start to explore sex as a biological variable in that way. Um, Another kind of major obstacle that we've encountered is actually studying sex differences in human tissues. So again, accessing human tissues is extraordinarily difficult. We rely a lot on public resources and public databases to procure human tissues for research. These cost a lot of money and a lot of effort, but I think it's worthwhile but you have to kind of acknowledge that this kind of research takes a long time and it takes a long-term commitment. So I think that's another major challenge in doing sex differences research, especially with human tissues, is that you need to sufficiently power your analysis and have a sufficient amount of sample to be able to claim a sex difference in your data.
Thank you so much. Actually, now that you mention this, talking about your first point, I just started working with a new cell line, primary cells, and now I realized that when I was reading the specifications and the details, when it's about the sex, they don't mention, or either they mention they are mixed or not specified. So
Exactly. Yeah.
now is the first time I actually realized.
And I know there's active push to try to get companies and to update catalogs with information about biological sex and ancestry of these cell lines to help researchers really figure out, is their phenotype dependent on any variable that might be may have been unknown previously because of poor reporting.
Yeah, absolutely. Thank you so much. I would like to move forward with our third question for this interview, which is how do you envision your work on sex specific biomaterials contributing to more personalized or equitable cardiovascular therapies in the future?
This is a great question. And I think that it's one that kind of requires me to first define what is precision medicine, right? And precision medicine, I think, really lies on the spectrum, right? We can think of one size fits all approaches where ideally, that's the ideal, right? We want a therapy to work for everybody. But we know that's not the case. We know that 8 of 10 drugs taken off the market because of adverse effects in women, we know that drugs don't work the same in everybody, right? So the one size fits all approach to medicine doesn't really work. Well, it works in some cases, but not in all cases, right? There's always this exception of a subset of patients that didn't respond to a therapy or didn't gain immunity after vaccination, et cetera, et cetera, et cetera. There's so many examples of that. Now, if you go on the other side of the spectrum, all the way to personalized medicine, where we're talking about a sample size of one patient and designing a therapy for one patient, right? I think that that's also terrific. We want to help individual patients with their individual needs. We also have to acknowledge that those kind of treatments are most likely accessible to, let's say, the top 1% of patients, you know? And this also exacerbates an inequity, I think, that needs to be addressed. So my lab, we've kind of taken this approach where we're in the middle, right? Where we like to think about patient-specific variables, but stratify patients into reasonable categories that we can start to define uh, specific treatments based on a particular patient variable. In our case, we're focused on biological sex. And I think that that's possibly, arguably, one of the most impactful variables that you can account for. Because if you're talking about designing a therapy that only works for 50% of the population, if you just make a couple changes to your approach and make it, let's say you have a male specific drug candidate that works in 50% of the population in males, if there's a dosing strategy or maybe a combinatorial treatment that works more effectively in females, that's the kind of impact my work seeks to have. We want to show that we can just make subtle changes to dosing regimens or drug combinations, etc., to try to achieve a balance between a one size fits all approach, but also like not really going into the N of one precision treatments.
That's totally true. I agree that ideally we should be able to generate a treatment personalized for every person, right? That'll be like the ideal. But in reality, we know that that's not going to happen, or that is not going to be the reality for most of the people. And I think the approach that you are taking is the one that is going to benefit a larger community. So that's great.
Wonderful. Thank you so much. Yeah, it's really exciting time to be a part of this work.
Yeah, absolutely. Well, I would like to continue with our final question, which is for young scientists listening to this podcast, what advice would you give to someone interested in integrating sex differences into their own research?
Absolutely. So I think that there's this common misconception that by looking at sex as a biological variable, you'll be doubling your sample sizes, right? Now, that can be true in some scenarios, but if you choose the variables that you're looking at very carefully, um, you you can structure a study to perform a sex difference evaluation without really increasing the sample size or the cost of your study, right? So I think that's a common misconception. But I think like just a very quick way to get started is if you're working with cells or tissues or any sample, ask yourself the question, do you know the sex of that sample, right? I think a lot of researchers do not know. And I think it's a very low hanging fruit for an opportunity for researchers to start to ask themselves, like, what samples am I really working with? And is this cell line female? Is this cell line male? Is this tissue from a pig, a male pig? Is this heart from a female donor? Like I think there's so much to ask there about what samples we're currently working with and where our data are pointing us to, right? It could also be as simple as taking existing data and stratifying it by sex. Maybe you pooled your male and female mice together into one study when really you should be aggregating them based on sex, and maybe you'll see differences in how these male versus female mice respond to your perturbation, right? So I think that those are some very simple ways that you can start to integrate sex differences in your own research and start to use your already existing data sets or your already existing samples and just kind of do a small reanalysis pilot study. I also think it's important to kind of tap into your community of scientists, right? So, I mean, like I was lucky enough to have the kind of the right mentors to kind of support this kind of research direction. But I think that there's this growing community of scientists who are just as passionate about health equity um, that you might be. So I think it's just a matter of also tapping into that energy and attending conferences and learning about how people are incorporating sex as a biological variable in their studies, so that you can also be kind of a part of that culture change in science too.
Thank you so much for all of those advices. I'm also going to integrate them in my own research. And I think all of this is going to help all of our listeners. Also, something I would like to bring to the discussion, from what I have experienced, there is also a lot of misconception on sex differences because sometimes it's seen as a checklist of things that you need.
Mhm.
I think it's something that I have experienced, and instead of having just this checklist, we need to actually like design better experiments and have a better plan on how we can incorporate all of this information and what this data is going to provide us, like which kind of information. And I think that is going to help us to have more like complete studies that reflect better what we are trying to do.
Exactly. It's a matter of also looking at clinical literature to see if the particular disease you're looking at has a sex difference or not, right? If there is a documented sex difference, then you already have the blueprint to do the study, right? It's really a situation where it's like, let's look at the existing clinical data, and if a sex difference study is warranted, then we should do it.
Yeah. Thank you so much. I would like to thank you also for being here, for accepting to have this interview. I think it's really valuable, and this episode is going to be really useful for all of our community, all of our trainees listening, and also for taking the time to have the interview as well.
Thank you so much, Dayanara, for having me today, and I'm so excited to be a part of Beats research.
Thank you. So I would like to also thank all the Beats Research radio listeners for tuning in. Don't forget to like and subscribe to our YouTube, Twitter, and Instagram channels at Beats Research. And on behalf of our entire team and our director, Dr. Emilio Alarcón, we hope to see you all in the next episode.